So we'll be starting like 2-3 minutes. Let's start. Okay, so hope you had a great week and thank you for joining today also. I hope you joined last week also. So this is the second week live discussion for the tissue engineering course by NPTEL-PMRF program. So I'm Soumyadeep and let's start with today's discussion. This week, I hope you have gone through the assignments and what are the practice questions and what are the. I have seen some questions in the discussion forum. I would encourage you more to write more questions and have study more on this direction of not only just the material that is being provided. Study more in the when access Google Scholar and all those platforms to find more relevant information and ask the questions whatever you are finding. Okay. So this week we like week 2 we have studied about the what are the scaffolds and its role in tissue engineering, as well as what are the desired properties of the scaffolds. Its role is imparted by the scaffolds, that is cell-supporting nature, it should promote the growth, what are all these factors we will study. Then what are the different kinds of scaffold materials that are used, natural polymers, synthetic polymers, this kind of materials are used in the study about. Right. Then ECM, extracellular matrix and its components and various commercial ECM-based products that are currently in the market, and how they are produced and like decelerization technique and their various decelerization modes we have also studied. Right. And then various natural and synthetic polymers. What are the natural and synthetic polymers that are used for the scaffold fabrication? We will study about it. Then synthetic polymers and hydrogels and their components, how the hydrogels forms, cross-linking, various mechanisms of cross-linking. So this kind of mechanism you have studied, right? So we will be starting off with the question. It's a 3-part question. So the question says that X is a material, X constitute around 50% of the total body protein and is the most abundant protein found in human. It is predominantly synthesized by fibroblasts. It is primarily structural component of the dermis, where it provides strength and support to the skin. So based on the above explanation, answer the following 3 questions. Identify X. It's going to be an 8-letter word. You can use the chat box. I encourage you to engage in this answering. So, what do you think? What would be the material? So, it is the 50% of the total body protein. It is constituted of. So, what will be the protein? It is collagen. Yes, it is collagen, right? So, the next part of the question goes that X monomers self-associate into a what kind of structure? So, options given is single stranded structure, double stranded structure, triple helical structure, and D is amorphous structure. So, it is what kind of structure? Triple helical, right? So, it's a triple helical structure that is found. So, how many monomers are there? Like, what are the basic types of monomer in a type 1 collagen? Let's for example, how many? What are the number of collagen monomer that is present? 2 types. Yes, alpha 1 and alpha 2. Right. Right. Type 1 and type 1, type 2 is a type of collagen. Monomers are alpha 1 and alpha 2. Okay, Jati. So type 1, type 2 is the different kinds of collagen forms. Type 1, type 2, type 3, type 4, type 5. This kind of collagens we have, right? These are the types of collagen. The monomers are alpha 1, alpha 2, right? So, and they form a triple helical structure like alpha 1, alpha 2, alpha 1. So these three monomers come together and form a alpha helical structure. Okay. So I'd encourage whatever the questions you have, you just write it in the chat box. Okay, so that in case in the continuity, so others don't feel the problem. So the third question is the which of the following types of eggs, that is the collagen we have found, is or are the main component of bone and cartilage respectively. Bone and cartilage. Yes, option A, type 1 and type 2. Right? Type 1 and type 2. So these two types of collagen forms are main components of bone and cartilage that we have seen in the slides this week. Okay. Now, as our previous week's tradition goes, what is the different forms of collagen? That is type 1 to 5 we have. So scientists tried to check, like they thought, like if the same kind of collagen is found in the as in the normal tissue or normal cell ACM, is it the same for the cancer cells also? So they have checked it and they have found the case is a slightly different one. So they have found that the collagen homotrimer is formed instead of our regular collagen heterotrimer. So our form is like alpha 1, alpha 2, alpha 1. So it is a heterotrimeric form that we have in a normal cell. So they have tried with the myofibroblast cells. They have used it as a normal cell. And they have taken the pancreatic cancer cells and seen that the collagen type is different than the normal collagen type that is present. So it is forming a collagen 1 homotrimer, that is only the alpha 1 monomers are being used, and they are forming a triple helical structure and they are forming this kind of collagen 1 homotrimer. So how the cells, cancer cells, are achieving this? They're silencing the collagen 1A2. That is two types of genes we have: collagen 1A1 and collagen 1A2. These two genes produces this monomers. So they are silencing this collagen 1A2 gene by epigenetic modulation and thereby reducing the collagen alpha 2 monomer concentration in the cell. Their synthesis is regulated. Thereby, they're saying to the cells that form the more alpha 1 homotrimers. So, as these homotrimary forms are enriching the cancerous ECM, it is actually helping in the cancer cell proliferation as well as tumor organoid formation is also being encouraged. And this kind of alpha 3 beta 1 integrin interaction is encouraged by this homotrimer, collagen 1 homotrimer formation. And they have also found that the cancer cells microbiome, ECM microbiome, what the microbiota that are found around the cancer cells, it is also changing because of the formation of this collagen 1 homotrimeric form. Okay, so they have found that the anaerobic microbiota, anaerobic bacteroideals are being enriched in this kind of cancer cells. How they have found it? So what they have done is they have cancelled the activity of collagen 1 homotrimer, and they have seen that if this activity, this cancelling of this collagen 1 homotrimer, they have suppressed the cancer cells growth as well as T cell enrichment is also increased. T cell is response. So T cell enrichment is also increasing, and the microbiota is also changing. So the anaerobic bacteria, their control is very high. We cannot use any kind of regular broad spectrum antibiotic and try to suppress their growth or their growth. So we cannot do that with this kind of anaerobic bacteria. But if the collagen 1 homotrimer's activity is reduced, then their microbiome is also changing, and we can control that microbiome with the help of any kind of broad spectrum antibiotic also. So the main point is the different forms of collagen that is being enriched in different parts of our body. It is not stopping. Stopping there only. So the cancer cells are producing their own form of collagen. That is a homotrimeric form. There is only alpha 1 monomers being triple helical formed, and they are forming this homotrimer form, and they are regulating their cancer cell behavior. Okay, so this is a very recent article, like only in June only they have published it. So these have given a new revenue to target the cancer cell therapy. Okay. So this is one aspect of this collagen synthesis. Right? Let's move on to the next question. So this is regarding a glucose responsive hydrogen system, right? So the question goes: this glucose responsive hydrogens can act as an artificial pancreas to control blood glucose concentration. When the glucose concentration is high, the glucose diffuses into the hydrogen and is converted to gluconic acid by glucose oxidation. What will make what will happen to this pH after this conversation? The conversion that leads to the hydrogen swelling and insulin release. So, what will happen to the pH after this conversion that will lead to the hydrogen swelling and insulin release? So, the options are pH will increase, pH will decrease, and pH remains constant. Okay, okay, we are getting the response. We want the same pH will increase. I would encourage you to read the question once again. So, this part, when the glucose concentration is high, the glucose diffuses into the hydrogen and is converted to gluconic acid. It is forming a gluconic acid. Yes, pH decrease, acidity increase, right? So, it is forming an acidic form, right? Glucose to gluconic acid. So, yes, the correct option would be pH will decrease, right? Now, this part of the question says that the pH decrease has an effect with the hydrogen swelling and insulin release. What do you think what would be the mechanism for this? What do you think how this pH decreasing is helping hydrogen swelling and insulin release? What do you think? What is happening there? Just think about a hydrogen block. You have something like a block of hydrogen, and the pH of the system is decreasing somehow. And I am proposing that the water uptake is being increased and the carbon release is also increasing. How? Like how the pH decreases? Like, yeah, water intake maybe increasing. How? How the swelling and water intake is. Yeah, swelling is happening. That means water intake will be increased, right? Now, what would be the mechanism? What do you think? What can happen there? Just guess. Just try, like guess what might be the uh, change in cost of all right. Interesting. So you guys think that this is be the mechanism due to the diffusion and change in crossing parameters, right? These two options I have got. Okay. Anyone else would like to try? Just guess. Wild guesses. No problem. Okay. Let's see what is actually happening. So this glucose sensing mechanism, it is like 1980s, 1990s mechanism. They have been doing this. So mechanistically, what happens with this enzymatic system? There is the left portion of the slide. So these are the enzyme-based glucose sensing insulin delivery hydrogens. So what it actually does is this glucose oxidase enzyme is converting glucose to gluconic acid, correct? And they are reducing the pH. Now, the polymers that are being used that has a form of tertiary amino groups. It's a tertiary amino group enriched polymer. And because of the pH decrease, the amino groups, the tertiary amino groups are getting charged. It is getting cation. So, the whole charge density is also increasing of the hydrogen system as the glucose is entering. And as the number of tertiary amino groups are increasing, the charge density will also increase with as many glucose molecule are getting inside, because glucose oxide is doing its job. It is converting the glucose to gluconic acid and thereby reducing the pH, and the pH reduction is charging the polymer. So as the polymer is getting charged, the hydrogen, that is the hydrogen block, if you think about it, hydrogen block is getting charged. So the other form of The counter ions, that is the anions, would also try to come to reduce that charge because our system wants the neutral system, right? So the anionic concentration or the counter ion concentration increases inside the hydrogen or around the hydrogen system. So this high concentration of this counter ions, this leads to an increase in the osmotic pressure. Right. So more amount of water and more amount of counter ions will increase and their osmotic balance is changed. And this leads to the uptake of the hydrogen with more amount of water. So think about it. It's getting charged, more amount of counter ion is getting inside. The osmotic balance is changing of the hydrogen. So the water is rushing inside the hydrogen to get into to balance the osmotic pressure. Thereby the water uptake is increasing. And as the water is getting inside it, the pores are being filled, so that cargo release is also increasing. So this is the classical mechanism with the enzyme-based glucose sensing insulin delivery system. This is a classical mechanism of mostly the glucose oxidase enzyme is used for this kind of materials, and this is how the insulin delivery is regulated. Now, there are various challenges with it also. So, as these are the proteins, there are problems with protein degradation also. So, we are not very much, these proteins are not that much stable to be used in harsh conditions. So, in our body also, we have proteases. So, these proteins will also degrade. So, this is not a sustainable system to work with for a long term use for the insulin delivery. So, they have been searching for the solution where there is not that much use of this kind of living things, proteins, that is that much degradable. So they are searching for a synthetic alternative, and they have found it. They have seen that the phenylboronic acid based systems are one material that can work as a insulin delivery or the glucose sensing material. And this glucose sensing property can be used for the insulin delivery. Okay. So they have called it a smart gel, where they have used this kind of phenylboronic acid, this phenylboronic group. So it is like a phenyl group and two boronic, this BOH, OH. So when the glucose goes in, these OH groups interact with the glucose's 2OH group, and they entrap the glucose. And this mechanism creates a negative charge. So their anionic form from phenylboronic acid to neutral form, the glucose interaction changes the neutral to anionic form. So our target is achieved. So we have created a ionic disbalance in the system. So what the counter ions will do? Again, the osmotic pressure and swelling and cargo delivery. So they have used these phenylboronic groups with various polymers, polymeric synthetic polymers. They have cross-linked with this phenylboronic acid groups pairing polymer monomeric form, and they have cross-linked them from the hydrogen. And they have seen when the glucose is not there, it is in the collapsed state or the hydrophobic state. The polymer chains are entangled. Now, as the glucose is entering, the phenylboronic group is interacting with the glucose. Okay, so this interaction is again reducing the ionic imbalances created, and the osmosis and carbo delivery is happening with, and as because of this interaction, the polymer is also releases the carbo. Okay, so this is the entirely synthetic. No protein is used. This phenylboronic based system, smart gel systems are used for the insulin delivery. And they have created a smart catheter based system for the insulin delivery also, where they have used same kind of gel and coated it with the catheter system. And they have used this kind of material for the insulin delivery or cargo delivery. Or glucose sensitive cargo delivery can be done. They have used insulin as a cargo. But you can tailor it and you can search different kind of cargos and they use for this delivery using the glucose concentration. Okay, so this kind of glucose sensing mechanism is also there. So one classical system with glucose oxidase enzyme and one entirely synthetic system that is in the right hand corner where phenylboronic group like chemical systems are used for insulin. Okay, so this is the systems that are used for the glucose sensing mechanism. Okay, let's go to the next question. Which of the following statements are correct? The select all that apply. The options are pore size can be controlled during salt leaching process. B is gas foaming can be used to obtain interconnected pores. C is while performing gas foaming, there is less control over the pore size and distribution compared to salt leaching. These three statements. We have studied in this week this porosity, how the porosity is increased in the or reduced in the hydrogen system. Remember for that? What would be the correct options? These two techniques, like salt leaching process and gas bombing. Yes, yes. Got a response. Anyone else? You have studied it, how the pores are being created, gas bombing, salt leaching, these techniques, interconnected pores, sparse pores, regularity, pore regularity. These things we have studied, right? Yes, I have got the response, and all of the above statements are true. Pore sizes can be controlled during salt leaching process by increasing or decreasing the salt concentration in the blend. Gas foaming can be used to obtain interconnected pores. Yes, that is true. And while performing gas foaming, there is less control over the pore size compared to salt leaching. Okay. All these things are true. So I want to talk about this biomimetic anisotropic hydrogen. Why? I'll tell you. See, what are the different kinds of hydrogen we are studying about? These are all regular in nature. So this synthetic polymer based or natural polymer based hydrogen that we are synthesizing, if you cut it and characterize in a particular angle, their properties would be same in every direction. But what are the systems that we are finding in vivo or in nature? They're not always isotropic or they're not always regular. We need to find certain solutions by which we can create this kind of irregularities. So, in scientific terms, there are two different kinds of hydrogens that might be there. One is an isotropic form, and that is an anisotropic form. Whatever the synthesizing we are hydrogen synthesizing with the help of natural polymer or synthetic polymers, we are mostly synthesizing this isotropic form. They are regular in nature, their characterization parameters, everything is regular. So if you cut it and characterize it, their properties will be the same. They will not change with the angle of their characterization. But our natural system or indigo system is not this much regular or isotropic in nature. They are anisotropic. So there are different kind of structures that are being created inside the hydrogen-like system. So these structures have very important role in the control and cell development and their cell differentiation, their migration. Everything is controlled depending upon the anisotropy of the system. That's why this biomimetic anisotropic hydrogen systems are being created. So along with this isotropic hydrogen that are regularly being used, anisotropy is being induced inside the hydrogen. Okay. For example, in this diagram, in the left side, A. So what they have done, they have created fiber-like structures inside the hydrogen by the mechanism where they have taken a hydrogen block, isotopic hydrogen block, they have fixed it, clamped it, and then tried stretching the hydrogen. So what it resulted is fiber-like forms are produced in the parallel direction. Okay. So this and they have used this kind of anisotropy that is being created and layered it, or sometimes use that only and from you can see in this orthogonal lamination or axial rolling or concentric or countercentric rolling, depending upon various mechanism of using it, they have used this kind of anisotropy and formed various anisotropic hydrogen systems. And these anisotropic hydrogens are biomimetic. Biomimetic means our natural biological systems contains this kind of anisotropic, and producing this kind of biomimetic anisotropic hydrogens are giving the scientists the more in vivo-like environment that we have been studying about. So our basic interest was what? We needed to create a in vivo, more in vivo-like system. That's why we're studying about ECM, the natural polymer, synthetic polymer, because we want to create or give the cells an in vivo-like system. So this kind of biomimetic anisotropic hydrogen systems are being used, and they have this kind of structures, anisotropic structures, and they are giving the cells the environment that is much more in vivo. Right? So, for example, they have also created this kind of blood vessels, anisotropic blood vessels for the delivery. You can see in the C, figure 4C, anisotropic blood vessels are also created. So, whatever, if we are using a simple pipe for the delivery of fluid, our blood vessels are not like that. Their structures are highly irregular, and each and everything has a role for it. These irregularities we need to take into account also. Not only just creating a regular structure, a hydrogen block or a regular structure is not going to give the cells as much amount of irregular environment. We need to take into account this anisotropic and create this kind of biomimetic anisotropic hydrogen. I just wanted to introduce you to this concept and a brief example of how this kind of structures are being created. You can get into much more details. I have given the source also. The slides are also. You can copy paste the TOI and follow the article regarding how this kind of biomimetic anisotropic hydrogens are synthesized and what are the various kind of use that it is. Okay, let's go to the next question. Question says the extracellular matrix is composed of proteins and proteoglycans. Proteins can be subclassified as structural and specialized proteins. Which among the following are structural proteins? Select all that. Options are given: collagen, laminin, fibrin, elastin, and fibrillins. What do you think? What would be the correct answer? Which among these options are the structural proteins? We have studied about it. In the ACM section. We have studied in the ACM lecture by Professor Vignesh. We have studied this. What are the structural proteins? We have any response. What do you think would be the structural proteins? Okay. Select all that apply. Like what are the options? Right. Yes. So the correct option is A, D, and E. That is collagen, elastin, and fibrillin. These are the structural components. The laminin, fibronectin are specialized components. They are glycoprotein. So, correct option would be collagen, elastin, and fibrillin. Well, from this glycoprotein. So, what do you think the difference between proteoglycans and glycoprotein? These are two different things. Proteoglycans and glycoprotein. What do you think? And proteoglycans? Yeah. So, glycoprotein they make up the cellular membrane. They are present on the cellular membrane. So, what would be the structural difference between the proteoglycan and glycoprotein? So the proteoglycans have a protein core with the uh, carbohydrate structures associated with it. Right, one or more carbohydrate structures. There are long chain carbohydrate structures that are associated with the protein core. Carbohydrates, what are the specialized carbohydrates? Glycosaminoglycans, GAGs. So those are the proteoglycans. Okay, and the glycoprotein is proteins with carbohydrate. It is covalently attached with the glycoside. It doesn't need to be essentially GAGs. It can be any kind of carbohydrate. But these glycoproteins are proteins with glycosidic bond linked carbohydrates. Okay, and proteoglycans are GAG linked, and the protein core as a length of protein, and there will be different glycosaminoglycans attached to the structure. Okay, this is the basic difference of the proteoglycan and glycoprotein. That's why I wrote glycoprotein here to make the reading of this. That there is a difference between this proteoglycan and glycoprotein. So we have talked about decellularization and the ECM center. So ECM decellularization techniques can be with the help of either chemicals or physical mechanisms or enzymatic mechanisms. So there are various kinds of mechanisms associated with decellularization. Mostly decellularizations I want to talk about is two kinds of decellularization application depending upon where they are being employed. So decellularization can be of two types depending on the type of application. So it can be either ectopic implantation or orthotopic implantation. These decellularized matrices can be used either ectopically or orthotopically. So what are the difference? Ectopic means we are repurposing an ECM from a different site to a site of our interest. And it is like it is not the same site of the these two sites that we are using is not the same. While the orthotropic system or orthotropic implantation, we are using the same sites ECM and repurposing. So there are advantages associated with the orthotropic implantation because we are using the same site or same ECM component. The cellular interaction is much more it's better in case of this orthotropic implantation because the ECM kind or the what are the mechanical stimulus or what are the mechanical or chemical information that is held by the ECM is same because it is harvested from the same site. While the ectopic implantation is repurposing, they are using one ECM from other side and then using it to the other purpose. Right. So examples of this orthotropic reconstruction goes like they are mostly used for the hard part. So for example, if we think about is like we are taking a porcine hard part, decellularizing it, removing all the cellular components, and then reseeding with the patient's cell, culturing it, and then implanting it to the patient. So that is the mechanism of this orthotropic implantation. Why? If we take like a porcine skin and then repurpose it or seed it with the porcine skin ECM and then seed it with cartilage cell or various specialized cells and then use it for the seeding of this with different kind of cells. That is what we're taking the ECM cells, ECM from the dermis and then using it and seeding the cartilage cell and using it for our purpose. Then that is a ectopic implantation. Okay. So these two different kinds of mechanism is happening with the decellularization. Okay. So why I started talking about this is because this decellularization can be done by either this decellularization that is saving a different kind of cells and then implant it to the site of our interest, or we can just directly implant it in the site of interest, or we can we can modify the ECM. We can blend it with other kind of synthetic or natural polymer, and then provide or functionalize it our ECM, and then use it with the site of our interest. This combined with the natural or synthetic polymer and refunctionalization of the ECM is now being studied, and they have found that the properties that might have lost because of the decellularization technique that is being used is being currently it is helping to regain those kind of functions that might have been lost due to the destabilization. Okay, and in case of orthotropic reconstruction or orthotropic implantation, cornea is specifically hard to get the uh, ACM from and using because due to the digitalization techniques, the transparency that is required for the corneal implantation is lost most of the time. But scientists have shown that reseeding with the patient cells and after growing the cells for a few days, when the cells start secreting their own ECM also, so that have regained the transparency of the construct. They have used it for the implantation. Okay, so we have to keep in mind so two types of implantation techniques: ectopic and orthotopic. And what are the not only just the ECM as such we are using, we can also blend it with our natural or synthetic polymer solution or polymer component to regain the purpose of whatever might have been lost and we can use it. For example, this kind of blending is like incorporating a anticoagulant in the ECM to stop the blood loss in the blood vessels. So these kind of examples are also there where refunctionalization or functionalization of the ECM is also there. Okay. So let's go to the next question. The next question is RGD is a cell surface receptor binding domain of fibronectin that binds to what? It would be a 8-letter word. What do you think? RGD is a cell surface receptor binding domain of fibronectin that binds to what? What is the protein that it interacts with? Anyone? The RGD domain of fibronectin interacts with what of the cell surface protein? Yes, yes, it is the integrins. Integrins are the integrin is the right integrins or integrin, whatever. It's an 8-letter word integrin. Answer. Integrins also is. So the RGD or the fibronectin interacts with integrin. Now, why I have put this question? I wanted to talk about integrins with a little bit more detail. Integrin is the kind of cell surface protein that is ubiquitously expressed in all metazoan cell types. In all the cells that we see has this kind of integrin to interact with the fibronectin or the interacting domains of the ECM. So, and this integrin has the function of like cell adhesion, migration, proliferation, survival, metastasis, mechanosensing, cytoskeletal localization, everything. Integrin is important for all the cellular function that we can take about. Now, how this interaction or integrin binding with the RGD domain that we have studied, like fibronectin has a RGD domain, that is the triamino acid domain, that interacts with the integrin that we have studied. So, how these things happen? This happens by two mechanisms. This interaction. This can be by two methods. We call it a bidirectional signal. So it can be inside out signaling or outside in signaling. Okay. So what are to do this? So the inside out signaling says the internal signal element binds to the cytoplasmic site. So this integrin is like having a outer like outside the cell region, a transmembrane region, and inside the cell region. So it spans the whole thing. So the cell membrane, it has the outside part, there's a transmembrane part, and inside the cytoplasm. That is the cytoplasm. So this internal signaling elements bind first with the cytoplasmic part. This is the inside out signal. So the inside of the inside the cells part is being interacted, and this induces a structural change, and this activates the integrin. and this integrin then interacts with the binding proteins. And the reverse is true for the outside in signal. So the outside signals it activates the integrin, it structure change occurs, it interacts, and then the downstream signaling elements are being activated with the help of this cytoplasmic. Okay, so these two mechanisms are required for the integrin functionality. And not only just fibronectin, there are other proteins which also uses this RGT-like interaction domains. For example, this I have given here petronectin, fibronectin, laminine, inactivin. So there are various other protein also that has these RGT motifs that helps in integrin interaction. So don't think that the fibronectin is the only protein or only component that integrins interact with. Integrin is adhesome is adhesome is called. So this adhesome part, integrin's part is much more higher than the fibronectin. They interact with a lot more components, not only just the fibronectin. Okay. Another thing is we might think that if RGT domain is there, this protein is interacting with the integrin. That is also not the case. What happens is sometimes the RGT domain might not be accessible for the integrin interaction. So this interaction occurs, you can see here. This interaction domain, this is the part where the RGT motif binds. Okay. This beta subunits part where the when it is activated, it is this part is activated and this interacts with the RGT domain. Okay. Now the RGT motif has to be interactable with this part of the integrin. Okay. So if the RGT motif is not accessible by this integrin, so this RGT motif, having RGT motif is going to perform no good. Even if you think that it is true for the larger proteins where the tertiary secondary structure is hiding the RGT, it's not the case. So there are smaller like hexapeptides or this kind of smaller blocks also where RGT protein is somehow not being accessible or not being represented in a correct way so that the integrin can act. So this think about it this way that RGT is a motif that is responsible for the integrin interaction. Good. But having the RGT domain in the protein does not necessitate that the integrin is going to interact with the protein. Okay, it needs to be accessible and needs to be presented to the integrin in the proper form so that the integrin interaction can occur. And the downstream signaling for the elements activation and whatever the downstream molecular pathways and whatever the function the integrin, the myriad of different function that is being performed by the integrins can be performed only by presenting it in the correct form and presenting it. Okay. So that's the integrin part. Okay. Now, next question is we have studied electrospinning, right? So, electrospinning, the cone-like structure forms during electrospinning is known as the cone-like structure that is formed during electrospinning is known as options are given Garner cone, Baylor cone, Taylor cone, and who does not form. Yes, I have getting responses. What responses here? Yeah. So the correct option is Taylor cone. Right. So the cone that is formed at the top of the spinner is called the Taylor cone. So how this cone looks like? This conical structure forms when there is a huge potential difference. It is in the kilovolts. Mind it. It's not like our wall plug is 220 volts, right? It is in the kilovolt range. That much high voltage potential difference is created to make this kind of conical structure where the components, the individual the building blocks of the solution that is being electrospun, it starts repelling each other in such a way that it starts to form a cone-like structure and gets ejected out. It started whipping in the form of after forming this cone when this polymer solution is injected, it starts with the whip and gets collected in the surface. So if you think that the spinneret is or the it can be only the needle for electrospinning. It is not the case. The spinnerets that is used can be of different form. So classically it can be a single needle form where the needle top is being used for the charge differentiation, or it can be also a multi-needle form, and sometimes it can be coaxial form also. So coaxial form, what it does is one polymer solution is getting ejected while another line, another polymer solution is also being fed. So that what it does is one polymer core and it is inserted by the second polymer. Okay, so this the middle part that would be created with the core. This part, this needle, and this second one, this second needle is creating the encircling layer. So these core shell kind of nanofibers are created with this kind of coaxial spinnerets. Okay. Now, not only just that, we also have a needle-less system where this kind of cylinders or balls or disc spinnerets and various different investigation is being going on to create this kind of needle-less system. I have given a video here, so you can see this electrospinning is being performed from this kind of cylinder. So a cylinder is spinning with a multiporous cylinder is there. A polymer solution is being ejected and the charge difference is created and the electrospinning is being performed. The polymer solution starts putting out from this hole. So this kind of cylinder-like spinners are also being used. These are needle-based system. What are the advantages it would provide? There would not be, so needle-based system has a problem in needle clogging. The polymer solution may get dried up in the needle only, and the whole electrospinning process will stop because the polymer solution will not be able to eject now. So how to resolve this situation? We need to create certain situation where there is no needle is used. So there is no needle clogging. Another thing is you can see the how much large amount of uh, fibers are being spun out at the same time. So this form of needleless systems are being used currently to make large amount of fibers in a very small number. So the bulk production of electrospun fibers are this kind of needleless systems are being investigated. Okay. Now you have read about the static collector and rotating mantle. Collectors are also changing. So not only just a static collector or a rotating mantle is there, there are different forms of collectors. You can see there is a representation. Depending upon the change in the collector form, where the potential difference is greater than the electrospin fiber is being collected, the nanofiber morphology as well as the deposition pattern is also changing. So it can be either uh, random non-open state or it can be like parallel deposition of the fibers. Not only just that, it doesn't stop there. Depending upon the change in the collectors, the nanofiber morphology also changes. Okay, so there are various forms of collectors like this spinning disc collector or this rotating mantle. So this rotating mantle is not only just rotating in a single axis. The rotation is also changing and the Z movement is also changing. So these kind of dynamic systems are also being investigated and with different kind of morphology in fiber deposition pattern, and their functionalities are also changing depending upon this spinneret change as well as collective change. So there is a beautiful article at Chemical Reviews and electrospinning and various modification of electrospinning. I would suggest you go and read that article. It is a very big, like a book, kind of like 8 pages of uh, review article where they have shown all whatever the development, the history from the historical perspective to the whatever the recent trend that is coming up. So this kind of reviews I would suggest you go and read to get to know more about this kind of electrospinning mechanism, whatever the changes or whatever the advances that has been brought upon. Okay, so these are the 6 questions I wanted to discuss today. I am willing to take the questions now. Whatever the questions you have had, I have turned on the microphone also. I will go to the chat. So yeah, I have had a question that your lecture was very explanatory and interesting. But I wanted Related lectures. Okay, okay. So yeah, it is unfortunate, but yeah, these videos are being uploaded to YouTube. Whenever you find the time, you can go back and watch this video. If you have any questions while watching these videos, you can write about those questions in the discussion forum, and we can discuss. We can answer those questions. Okay? So I would encourage again that use the discussion forum, interact with the TAs, ask as many questions as you wish. Okay? So yes, uh, try to join, and if you are not being able to join, then go to the YouTube sessions and watch it. But it would not be this interactive. You are not really able to answer the questions and ask the questions, okay, at the same time. Okay, so the sample question answer would be provided if you would be it would be given in the slides only. So answer is there in the slide, and it is why the sample question is given that to make you think that what would be the potential answer if you are wrong. Go back to the slide. So if you read more, but if you find that yeah, you are confused about it, want to talk about it, I suggest you take the question and write it in the discussion forum. We will discuss whatever the question you were confused about. You think that might be the different answer that you were confused about the options. Okay, we can discuss there also. What is the channel? So yeah, it is given in the NPTEL site page where it is in problem solving sessions. So it is under my name. So the link is given in the problem solving session section in the NPTEL page, channel page. Provided. All right. So any other questions in the week's content or today's session? Any other question you have? In the meantime, so what we have studied today, we have uh, given a brief introduction or a brief discussion about what we have learned into this week, and we also discussed various topics from this simulation to electrospinning, from human biology to cancer cells, one time of formation, and what are the various kind of research techniques and the atomic and other modes of evaluation. So these are the things that we have discussed today. I will encourage to go to the video and see if you have any questions when you see watch it again. Okay. And the slides are also given in that problem solving session link only. So you can find the sources, what are the websites or what are the articles that I have used. Go back to those articles and read about it if you want to know much more about it. I have tried to grasp the information and give it in a much more concise way. So if you want to know more details about it, I would suggest you go to the those sources also. Okay, yes. Research article reading is kind of a habit. So when you first start, you might find that it is the topics are not that fascinating, and you are finding it not that much engaging as you can find in a book. So research articles are directed to let the journal subscribers or let the people know that this discovery or this information the group has created, researchers have created, and their only purpose is to let the information out. But when the book is being written, the book is written with the idea that the information needs to be captured by the students. The people who are reading it, they need to remember it, they need to find it enjoyable. The information need to be much more concise, and we don't need that much of the detail of the book for writing the book chapters. So that's why there might be certain issues when you are reading the research articles. That first initial stage. But after you have studied, you keep trying as much, like you try to read as one article, not just one article a day. You can take your time, read the article. You can sometimes leave the material methods section first. Now the most of the major journals, what they are doing, introduction, then results, then after that they are coming to the materials. Okay, so for older papers, I would suggest that you would study the introduction, then the result section, and then the discussion section. Or introduction, discussion, then the result section. So whatever you choose, you read and try to read it and take your time, read it slowly, try to understand it, and then you go to the material analysis. If you want to study about how what are the techniques and how they are got the results. And inevitably, when you are studying about the results, you would think in the back of your mind about what are the techniques they have used to get these results are. How these beautiful images coming of the hydrogen. So it is scanning electron. Let's see how what are the parameters they have used for the scanning. So that's how you go back to the material methods and then try to get it into the system, the habit of reading the research articles. And review article is like the book chapters or like the book article. So it is targeted to audience where they need to grasp the concept and the more concise and presentable information has to be given. So the review article also, I would suggest you read the review articles. Take your time, read it slowly, and try to. And you don't need to remember. You can always go back to the article and study it again and again. You don't have to mark it up. Just read about it, understand it. It will automatically. Okay. Alright. So with this note of you going back to the discussion forum, ask as much questions as you wish and interact with the TA. I would end this session today and we would see you in the next. Okay. Thank you. We can end the session.